Our next speaker is Dr. Brian Freed, who is currently a professor of medicine and immunology here on campus, as well as the executive director of cell gene therapy. Dr. Freed received his PhD from Albany Medical College, and that's exactly where he started his career as an assistant professor. He was recruited here to our campus, I believe, in 1997 as an associate professor of medicine and immunology. And he has continued on here on campus. Today, Dr. Freed will be talking to us about HLA gene editing to treat refractory rheumatoid arthritis. So Dr. Freed, welcome.
Thank you. So this is, of course, a very experimental system that we've been launching over the past 10 years. And so very happy to present this to you and get your feedback on this. But this is the HLA locus. It's on the short arm of chromosome 6. If you're a mouse immunologist, you don't really think of how polymorphic this system is, but there's really 9 different genes in here. I'm only going to be talking about one of them today, DRB1, which is made up of this chain here, DRB1, with an alpha chain that's not polymorphic. But the DRB1 chain itself is so polymorphic that there's over 2,000 alleles, as you can see here. Now, when we say there's 2,000 alleles, that means in the population, that's how many we've mapped so far. And that number has not slowed down. So it's probably going to be much, much larger than that, in spite of the fact that only about 100 of these are common. So rare alleles are actually common. In fact, rare alleles, there are more of them than there are of the common ones, if that makes any sense to you. And of course, the purpose of these molecules is that they present peptides. You know, we think of them as viral peptides of roughly 10 to 14 amino acids, and that's what the T cell sees. So the T cell can only see peptide presented by an HLA molecule. And so, of course, this incredible variability is there to protect us as a species. If some virus comes along that somebody can't present, they're going to die from that virus, but somebody else will survive from it. When we look, of course, in some cases, we have autoimmune diseases where we're presenting self-peptides. And in this case, in particular, I'm just going to talk about one of the many that you really can't see up here, but that includes type 1 diabetes, ankylosing spondylitis, MS. I'm only going to talk about rheumatoid arthritis, but you can see from this GWAS study that the association of the DRB1 locus with rheumatoid arthritis has a p-value of 10 to the minus 581. So enormous significance that's typically not seen in medicine. With current mapping of we could call it fine mapping or high density mapping, this has really been identified down to the point of these 3 amino acids in positions 11, 71, and 74. So the polymorphism of the HLA molecule exists within this peptide binding group, not so much the rest of the molecule. And in these 3 amino acids in particular, a valine, a lysine, and an alanine in these 3 positions have a p-value now of 10 to the minus 600, so strongly associated with RA. But what's of particular interest is the fact that one of these amino acids, the middle one, position 71, if instead of being a basic amino acid, it's an acidic amino acid, it is highly resistant. So kind of looking like a light switch of on and off in terms of susceptibility. And now if we compare the susceptibility to RA with the susceptibility to severe RA, there's a strong correlation. So what we're really saying is that the alleles that are susceptible make you also susceptible to severe RA. And the R squared on this is 0.98, so it's almost a perfect correlation. So that, again, that one epitope, the DKA, which is really only found in 0401, is the most sensitive, the most significant. And then you see there's another group over here, 0404, 0405, 0408, 1001, also just about the same level of significance. And then down here, less so, is a 101 and a 102. Now, collectively, all of these alleles shown in red are what commonly referred to as the shared epitope. We've said for the last 40 years, this shared epitope is associated with RA. But in fact, we should divide it up because they're not all equal. And in fact, a 102 is really not at all susceptible to RA, so it shouldn't even be on this list. And then if you look at all of these in terms of severe RA, in fact, you would really only find 0401 and 0404. The others are relatively rare, and they're not really going to have much of an impact here. So that really says that we're focusing primarily on one or two alleles when we're talking about severe RA. Now, what is severe RA? And this really, we're talking about two different things here. One is severe, one is refractory. When we're talking about refractory RA, there's no really universally accepted definition, but it's basically patients who are resistant to multiple disease-modifying agents, including one biologic. Severe RA is referring to damage to the cartilage and bone, but approximately 10% of the patients who have RA fall into this category. So we're talking about the 10% that are the worst of these patients. And it's not that it takes 30 or 40 years to determine that. In fact, the absence of an early clinical response to the first biologics is indicative of these patients being refractory. But the point for this talk that I want to get across is that 50% of these patients are going to express the 0401 allele, which has this VKA epitope, and then 30% are going to express a few other alleles. But we're really going to be concentrating on 0401 because it's the strongest and it's also going to be the most common. So, up until now, we're just talking about association studies, but we don't really have anything mechanistic. So here is the way we analyze this mechanistically, and that is to take a cell line, this T2 cell line, it's a lymphocyte cell line, that does not express any HLA molecules, any class 2 molecules. And we transfect this with the DR alpha and beta chain, so we can put in a single HLA molecule of our choosing that we want to study. In this case, I'm putting in 0401 or 0402, which have the two different epitopes. One is VKA, one is VEA in those three positions that I talked about. And then we can measure the binding of a peptide that is thought to be associated with RA, and that is collagen. So we can measure the binding of a biotinylated collagen to this cell by flow cytometry. And then in addition, we can do site-directed mutagenesis to change one amino acid at a time and determine what is specifically functioning here in terms of resistance. So if we do that, what you're looking up here at the top row are flow cytometry diagrams. If you see two peaks, the left one is just the cell without any HLA molecules, so that's background binding. And then when the peak shifts to the right, now you're seeing specifically binding to the HLA molecule. And so these are kind of lined up here on the top as weak to strong associated with RA. So you can see 0401 binds 13-fold more of this peptide over background. The next strongest one is 1001, that's 5-fold, and 0404, that's 2.3. And then you get down here to these others that are actually quite low. Now, the level of binding that is required to make a T cell, to actually activate a T cell, is about 1.4. So it would be 40% over background. That's kind of a minimum level. So, obviously this 0401 is very, very strong. It's shown graphically right here, just with multiple data points. And again, you can see the severity of RA is associated with a high degree of collagen binding. Now, if we compare to an 0402, this is a resistant allele, and you can see it really doesn't bind collagen over background. It's essentially negative. So what we can conclude here is this 0401, which has the VK epitope, strongly binds collagen and is associated with severe RA. And then the VEA epitope, again, just one amino acid difference, does not bind collagen and is resistant even to mild RA. It's completely resistant to severe RA. But in fact, these two don't differ by just one amino acid. They actually differ by 4 amino acids. I was only talking about that one particular epitope that was identified in association with RA. But in fact, these two differ by 4 amino acids. So what we really want to do is just do site-directed mutagenesis and change one amino acid. And so we're taking 0401, which has that VKA epitope, and changing it to K71E. So we're changing a lysine to a glutamic acid in this position. So that's the only difference between these two. Now, this is a new allele that doesn't exist in nature. And obviously, what I'm talking about going forward is gene therapy. Could we just change one amino acid and make the patient resistant? So now we're introducing the VEA epitope as a single change to the HLA molecule. And when we do that, here what you're seeing over here on the left is collagen binding. So again, there's the 0401 that binds collagen very strongly and is associated with severe RA. Here's 0101 that binds collagen much less. It's still 2.1, so it's enough to stimulate T cells, but it's much weaker. And is not really associated with severe RA. It's actually associated more with a milder and sort of late onset RA. And then down here you see the resistant 0402 allele that doesn't bind collagen. And then finally our 0401 with the single point mutation. And so basically this one amino acid change turns 0401 into a binding profile that looks like 0402. So it's basically resistant. It doesn't bind any collagen. Now, if we compare influenza is a control in this experiment. In this case, the 0401 did not block influenza binding. In fact, it's actually stronger. So if you look at the reason for this, this is the collagen peptide that we're talking about. In position 9, it has a glutamic acid that interacts with that basic lysine, so that's why it binds so well. If we change this single amino acid to an acidic amino acid, now it repels this, and that's why we lose binding in right here in the 0401 K71E. Now, the influenza peptide does not have an acidic or basic residue there. It has neutral amino acids. So it's really not affected by charge. It's really just more a matter of size. And so all of these can still bind influenza to various degrees. So the point here is that we're really performing plastic surgery on the immune system. We're turning off the response to collagen, but we're not turning off the molecule entirely. Okay, so that's an in vitro study. Now we go to an animal model where we put 0401, 0101, and 0401 K71E into mouse models, so they're humanized mice. These mice in the proximal region is still mouse HLA, if you will. These are knockout mouse. These mice do not express class 2 on their own, but they do have the distal region, but the proximal region, but the distal region is now replaced with the human HLA molecule. That's what's going to bind peptide. But this allows this molecule to function in mice as in their immune system. And so, of course, there's several questions that we need to answer. First is, if we were going to do a bone marrow transplant like this, would it be rejected? Would that single amino acid be rejected? So what we did was we took a skin graft model, which is considered the most significant, the most severe, the hardest transplant to perform. So we take an 0401 mouse, and if we put an 0401 graft onto it, it takes, and you can see here at days 10 to 14, it hasn't rejected. And in fact, it goes on out to day 70, it doesn't reject. If we put on 0101, which is now a true allograft, it differs from 0401 by 10 amino acids. These all reject by day 14. That's what you see here. And then if you took K71E, now only differs by 1 amino acid. It does not reject acutely. And in fact, we take these out to day 70 and we put the skin on backwards so you can see it growing in reverse. And they do not reject. So the immune system doesn't appear to recognize this 1 amino acid change that we made. So then the question is, well, would it work? Would it do anything? Would it block rheumatoid arthritis? So we can't really induce rheumatoid arthritis in the DR4 mouse, at least not consistently, but we can immunize them and show a T cell response. So we take the same 3 mouse models, we immunize them with whole collagen and complete Freud's adjuvant twice, and then 30 days later, we take the lymph node cells and stimulate them with the immunodominant peptide of collagen and measure proliferation. So that's what we're doing here. So you see the 0401 mouse, this is background without stimulation, and this is with the immunodominant peptide of collagen. You can see a nice robust response. 0101 has a weaker response, but of course it binds less of collagen, so it's not surprising that its response is weaker. Oh, I'm sorry. And then finally, 0401 K71E, which really doesn't bind collagen, and there's no response. If we look instead at influenza, the same experiment, but with influenza instead of collagen, you can see again, robust response with 0401, somewhat less with 011, but also binds less of the influenza peptide. And now 0401 K71E, a robust response because it does bind the influenza peptide. So then you might be saying, well, okay, that's all well and good to prevent RA, but what about patients who already have RA and they've got these T cells that have already been activated? So this is what the T cell receptor sees. The T cell receptor sees an HLA molecule plus a peptide. So we have 4 clones of T cells here that are all specific for 0401 plus collagen. And you can see if you rest them, they go back to their quiescent state. And then if you increase the amount given collagen, you can see a nice robust response by IL-2 production in all 4 lines. If you give them the wrong HLA molecule plus peptide, again, the T cell is specific for 0401, they don't respond. And if you give it K71E, now that's the correct HLA molecule plus the peptide. However, this K71E has been switched to a glutamic acid, so now the peptide doesn't bind, and these T cells aren't activated. So basically, even an activated T cell that's floating around the body, when you get new antigen presenting cells, they won't function, they'll quiesce. So the target, what we need to do in bone marrow is to replace the antigen presenting cells. These are macrophages, monocytes, dendritic cells. They come out of the bone marrow. They generally only live about 3 weeks, so that if you do a bone marrow transplant, these cells become rapidly replaced. Now, the beauty of this is that these cells are going to only, with the K71E, they're going to lose their ability to present collagen. And so collagen reactive T cells won't be able to respond. However, it's not globally immunosuppressive. So if you look at all of these other agents that are currently being used in rheumatoid arthritis that globally suppress the immune system, we're not doing that. We're only blocking this very early phenomena and specifically blocking that's collagen reactive. And so if we do a bone marrow transplant, we take the 0401 mouse and put the 0401 K71E cells in there, and then we follow them out on days 28 and 56. And Mark would recognize that that's kind of the days you look at for bone marrow transplants. And then we can monitor these. We have probes that are specific for 0401 versus 0401 K71E. So, and I've color coded them here so you can see green represents the 0401, blue represents K71E. So in the 0401 mouse, it's all green. In the K71E mouse, it's all blue. And then if we take the green mouse and give them a bone marrow transplant from the blue mouse, now you can see by day 28, it's about 3/4 of the cells in the blood are K71E, and by day 56, it's nearly complete. By the way, we followed these out. We looked in spleen and lymph nodes and in various tissues, and these cells have now infiltrated everywhere. So basically, by doing this type of transplant, we can replace the cells everywhere. So how will this happen in humans? It's a little bit different now. In humans, we have to isolate the bone marrow, isolate the CD34 stem cells, and use CRISPR with a guide RNA that will specifically target the 0401 molecule. It will cut there, and then we can fix it. Now, all we have to really do is replace this AAG with a GAG. So it's really one nucleotide difference, which will change the coding from a lysine to a glutamic acid. However, if we do this one edit, you won't be able to see it. So instead, what we do is we insert a whole cassette, this is the repair vector, with the new gene of interest, the 0401 K71E, and we tag along this truncated nerve growth factor, and that allows us to find the cells that have been edited. And so by flow cytometry, if we just measure this CD271 binding, you can see that cells that from an 0401 individual, if we edit the cells, you can see here are the cells that have been edited, and they express the 0401 K71E. We actually isolated them and sequenced them. And an individual who does not express 0401 doesn't have these cells. So we can specifically edit the cells of interest. So how will this happen in a patient? Well, it'll start by a GCF mobilization from the bone marrow. Then by apheresis, we'll collect these stem cells and isolate the CD34 fraction. Then we'll do CRISPR on these, and I'll tell you a little bit more about this in a second. Isolating those cells that have been edited. And then we've been working with Terumo here in town for the past 10 years, growing hematopoietic stem cells in a bioreactor, so we can greatly expand these cells here for infusion into the patient. Now the patient, unlike the patient Mark was just talking about, these patients don't have any cancer, so we don't have to be so aggressive. We don't have to wipe out anything in the periphery. In fact, we don't even have to wipe out their T cells. So really what we need is a low-dose busulfan type therapy that will just create enough room in the stem cell, in the bone marrow, for our stem cells to take. So this can actually be done, at least in the future, as an outpatient infusion. It really would be a one-time treatment. We'd see the rapid engraftment. By day 10, they should really, kind of be out of the woods. And the other important point is they're not immunosuppressed. We haven't really hurt their peripheral T cells. In fact, this agent doesn't even suppress their peripheral T cells. So there's really no global effect. Now, I did mention, this CRISPR-Cas9 kind of just as a simple box. If you really look at it, it looks something like this. Obviously, from the FDA point of view, there's an enormous amount of testing that's going on. So this is really the whole issue. How can you do this safely and have the purity and potency of this? You haven't introduced mutations. So there's literally hundreds of questions that go on in terms of that isolation procedure with every step of the way having criteria for selecting these the cells. So I'm going to end there. I just want to thank a very talented group of individuals here, specifically Kirsten Anderson, who was a graduate student here and kind of developed this peptide binding assay. Really measuring peptide binding to HLA molecules is not as common as you think it is, and really perfected that, as did Tina Rourke. Tina Rourke has developed all of the CRISPR modeling that we're using. And Viva has done all of the animal work on this. And then we have collaborators who have helped create the vectors that we're going to be using for the clinical trial. And then beyond that, the specific, the scientific staff, we have a huge regulatory group as well that, we have, actually now we have 13 patents, we have 2 FDA licenses, we have an IND, we just completed a study of mesenchymal cells for diabetic foot ulcers. So, we have a lot of experience dealing with the FDA and getting these things through. We know what we're up against with gene therapy. It's not going to be easy to get this approved, but I really believe that this is the staff that can do it. So I'm going to end there and I'm left to any questions. Hopefully, Mark will have a new form of transplants that we may be doing to replace the ones that you're getting rid of.
Okay, thank you very much, Dr. Freed. Are there any questions?
This is Steve Druskin. 2 questions, Brian. Are you going to have this insert in your final therapy, or can you just follow collagen binding to say CD14, CD16 positive circulating cells?
That's still open. Right now, we're doing it because the efficiency of any type of CRISPR, we've gotten it up to about 15%. What we don't know is maybe that's enough. What we do find in RA patients is the presence of that BEA seems to be dominant suppressive. So it seems like you may not need 50%, 100% of the bone marrow being replaced. 15% might be enough. So in other words, you might even be able to just edit, put the cells back in, and that gives you your resistance. We don't have a great way to test that prior to clinical studies. The animal models, when we do the bone marrow transplants in the animals and then immunize them, we've also got to deal with the fact that they just got busulfan. So, there's more of a suppression going on. So it's really hard to test that until we get to the clinic.
So one other quick question. When I think of rheumatoid arthritis and pathogenesis, I'm not sure why, but for some reason I think about citrullinated peptides and not collagen. Has that changed over the years?
No. So, again, we didn't have, in interest of time, we didn't have the time to show that. So 0401 has an extremely strong preference for citrullinated peptides, because what you're doing is you're replacing an arginine with a citrulline. Arginine will not bind to the lysine, so they don't bind native amentin. But once you citrullinated, now the binding is very strong. So that's probably what's leading, and the same is true for citrullinated alpha enolins. So the most susceptible allele binds these citrullinated peptides very, very well. Extreme preference for it. So yes, we think that's important early on. The thing is, that could go on for like 10 years before RA develops, before it becomes overt. What we're thinking is later on, at the point that we're going to be involved, Now you've got, a later response where you've got an immune response to collagen coming in later. So yeah, that in the early phase, yeah, you're right. It would be the citrulline peptides, and we have measured all of those and published on them.
So Brian, why do you think that only if you were able to get 15% of the modified T cell or modified stem cells in there, why do you think that would be even close to sufficient?
I know. I don't have a good answer for that. There seems to be evidence, at least in type 1 diabetes and rheumatoid arthritis, that resistance is dominant, but we've never been able to come up with a good answer for it. In the case of diabetes, it seems like the DQ6 molecule, which can also bind hybrid insulin peptides, for example, induces regulatory cells. But regulatory cells seem to be very important in type 1 diabetes. And if you look at the like IPEX type phenomena where you get this, a loss of a regulatory signal in the thymus, and then you see this sort of loss of regulatory cells, and you see diabetes. You don't see typically rheumatoid arthritis. Now maybe you would if you waited long enough. But I think it suggests that maybe the regulatory cells are not as important in RA as they are in diabetes. So how that, negative epitope works, inhibitory epitope would work in that model, we don't really know. Don't really know.
So a second follow-up to that. So you say in the mouse, it's hard to look at this because you've given them bisulfate, so they're not responding to collagen.
Yeah, well, so we did a bone marrow transplant in those animals so far. That means 6 grays of irradiation. So that's the immunosuppressive. Now, when we do busulfan, maybe we can. Yeah, so now we're going to switch from 6 grays of irradiation, which is more globally immunosuppressive, to busulfan. Maybe we can detect it in that.
Okay, so you haven't tried that?
No, that's what we're doing now. We're really at the stage now. The FDA likes to be involved early, but if you get too early, then they'll say you haven't done your work. So you got to pick. So this is the stage we're going in with, where all of the animal data is done, and now we negotiate with them over what would be the appropriate clinical studies or preclinical studies needed to get the approval to actually do this in humans.
Thanks.